The first stage of the cell cycle is interphase. Interphase has three stages. They are G1, S, and G2 phase. In the G1 phase, the cell grows and prepares for DNA replication. In the S phase, the chromosomes are duplicated. The last phase of interphase is G2 phase. It completes the preparations for cell division by synthesizing proteins and growing. Prophase is the first stage of mitosis where chromosomes become condensed and the nucleoli disappear. Each duplicated chromosome appears as two identical sister chromatids. They are joined together at their centromeres. The mitotic spindles begin to form. The centromeres move away from each other due to the lengthening of microtubules. The next stage is metaphase. It is the longest phase of mitosis. The centromeres are at opposite poles of the cell. The chromosomes line up on the metaphase plate. Each chromosome is connected to a connector core, and the connector core is connected to a connector core microtubule, which are coming from the opposite poles in the cell. The third phase is anaphase. Anaphase is the shortest phase of mitosis. Anaphase begins when the cohesion proteins are cleaved. Each chromatid becomes a full-fledged chromosome. The two liberated daughter chromosomes begin moving toward opposite ends of the cell as their connector core microtubules shorten. The last stage of mitosis is telophase. It is coupled by cytokinesis. The first two daughter nuclei form in the cell. The nucleoli reappear. The chromosomes become less condensed. Mitosis, the division of one nucleus into two genetically identical nuclei, is now complete. In cytokinesis, the division of the cytoplasm is occurring by late telophase, so that two daughter cells appear shortly after the end of mitosis. As a result of mitosis, two identical cells are created. If they were human somatic cells, there would be 46 chromosomes identical to each other in each of the cells. Mitosis is important to the maintenance of the chromosomal set. It also produces two daughter cells every time it divides. The cells are genetically identical to each other, but also identical to the parent cell. This allows for growth and repair of tissue.
Meiosis reduces the number of chromosome sets from diploid to haploid in a cell. Meiosis requires two cell divisions, resulting in 4 haploid daughter cells, with half as many chromosomes as the parent cell. Meiosis is important because it provides genetic variation. The process of meiosis creates genetically different offspring from the parents, which creates evolution. Meiosis I is the separation of homologous chromosomes in the cell. During prophase I, chromosomes begin to condense and homologs line up along their lengths. Then a process called crossing over, which is the exchange of DNA segments by non-sister chromatids, takes place. Chiasmata are the points where crossing over occurs. The chromosomes slowly begin to move apart once synapsis ends. The nuclear envelope breakdown occurs, and spindles are formed along with centrosome movement. During metaphase I, homologous chromosomes have aligned at the metaphase plate, and microtubules have attached to the kinetochores. Anaphase I begins the separation of homologous chromosomes to opposite sides of the cell. The spindle apparatus controls the movement of the homologous chromosomes. Sister chromosomes stay together due to sister chromatid cohesion. The start of telophase I means that each half of the cell contains a haploid set of replicated chromosomes, with each chromosome composed of two sister chromatids. Cytokinesis begins and a cleavage furrow forms, which splits the cell in two, forming two haploid daughter cells containing 23 chromosomes each. Meiosis II is the separation of sister chromatids in two haploid cells, producing 4 daughter cells in the end. During prophase II, a spindle apparatus forms and the chromosomes begin to move toward the metaphase plate. Once metaphase II begins, the chromosomes have aligned at the metaphase plate as seen in metaphase I. The chromosomes are not genetically identical because of crossing over from meiosis I. The kinetochores of sister chromatins are attached to microtubules, and anaphase II begins with the breakdown of proteins, holding the sister chromatids together at the centromere. The chromatids move towards opposite poles of the metaphase plate. In telophase II, nuclei form and the chromosomes decondense. Cytokinesis, or cell division, occurs, and 4 daughter cells are produced, each genetically different from the others.
Meiosis and mitosis are similar, but also very different.
Meiosis has 2 cell divisions, and mitosis has 1.
In meiosis, 4 haploid daughter cells are produced and 2 diploid cells are produced.
Meiosis occurs in humans and animal cells, and mitosis occurs in all cells.
Mitosis is an asexual reproduction, while meiosis is sexual reproduction.
In meiosis, the number of chromosomes are reduced to half. However, in mitosis, the number remains the same.